Hi, and welcome to Chapter 4. In this lecture, we're going to talk about histology and specifically focus on some intro tissue stuff and some epithelial tissue. So you have to excuse the construction site behind me, doing some home renovations. Got my pilot headgear on to try to keep the dog distractions to the minimum. So the first thing we're going to do is just start to talk about some terms, and I know you've heard of these particular terms before. So what's a biopsy? Well, as you probably know, a biopsy is when they go in and they take a sample of tissue and then they look at it under a microscope. This is why you want to have some knowledge of histology. And when they look at it under a scope, they try to look for any abnormalities to try to see if there's any sort of disease processes. So a person who would do this would be a pathologist. Their job is to study disease. And a histologist is someone who studies tissues, because the study of tissues is histology. Now you remember from last unit, right? Last chapter, we talked about the different levels of life. Excuse me. We know that the cellular level is made up of chemicals, molecules. Atoms make chemicals. Chemicals make cells. We know cells are alive, and therefore cells are going to form tissues. So a tissue is just a bunch of cells that have a common function. When we look at our tissue types, there's 4 tissue types. So right now we're going to fill out the table in your notes, very basic, because we're going to delve into each one. But you'll notice that there's epithelial, that's the hardest one to say, connective, muscle, and you can say neural or nervous. So if we look at epithelial tissue, basic characteristics of this stuff. Well, if we think about epithelial tissue, one thing is it forms tight sheets. So the cells are actually anchored together really, really tight. So cells have tight junctions, they're held tightly together, and they're sheet-like. Other characteristics of these things is that they're avascular. That means they don't have a blood supply. Now, you find epithelial tissue on the outside of your body and the inside. So I always say covering and lining. So the general functions of this is to cover and line the body. So it's really important for protection and things like secretion. Now, connective tissue, as the name tells you, it connects, right, and helps hold everything together. So connective tissue is characterized by having usually scattered cells and a lot of stuff in between. And the stuff in between we'll get to add soon, but that's called the matrix. So connective tissue characteristics, few cells, lots of spaces, lots of matrix. General function, connect, support, hold things together. Now, muscle tissue, the main characteristic of muscle tissue is that it moves. So it has the ability to contract or to shorten. And when we look at, so general functions is going to be movement. And muscle tissue and connective tissue, by the way, are vascular. They have a blood supply. Now, not all connective tissues are vascular, okay, but 95% of them are. All muscle is vascular. It all has a blood supply. And then neural or nervous tissue, again, characteristics of that are there are cells called neurons, which I know you've heard of a neuron, and then there's these neuroglial cells. Now, or you can say neuroglial or neuroglial, it doesn't matter to me. There's more than one way to pronounce everything in A and P. And so general functions would be just control. And we'll be looking at each of these tissues in more detail. So your book goes into this matrix first. So I put it here in my notes. Now, this whole matrix thing, you're going to really look at it in connective tissue. So it's super important in connective tissue. It's just the stuff between the cells. So if you have a cell, we'll put a cell here, we'll put a cell here, we'll put a cell here. Now, you guys probably remember from 119 or 120 that between the cells is all kinds of goodies. In fact, between the cells is fluid, so we call it IF for interstitial fluid. So this extracellular matrix is the stuff that surrounds the cells. It can be a fluid, a liquid, it can be a gel, it can be a solid, but it's on the surround cells, okay? Extracellular. So it's actually made by the cells, and this stuff is kind of like glue. It helps provide support and strength. It helps hold the cells in place, make sure they're going to the right areas. And it even has different chemicals to help maintain and develop, like some physiology things like mitosis and survival of cells. So when we look at this matrix, it's made up of 2 things. There's the ground substance and the fibers. So in my beautiful cell picture here, you saw my little cells, and this little yellow highlighted area, all of this stuff is the ground substance. It's the stuff between the cells. And again, like I said, We usually call it it consists of fluid called the interstitial fluid, or IF. It's going to have ions and nutrients, and then it has this like sticky stuff. So think of it as the glue. And the sticky stuff has big long names. You won't have to write them for me in a test, but you should recognize them. So we have our GAGs, okay? GAGs are glucose, aminos, glycans. So they're basically proteins and carbs, polysaccharides and proteins hooked up together. And you might have heard of chondroitin sulfate, especially it's a supplement. It makes up cartilage. Hyaluronic acid is another one of these. And these are just like the stuff, the glue between the cells. Another example is if you get a bunch of these proteins together and you kind of stick them all together, they form what's called a proteoglycan. So it's very similar. It's sticky proteins. And then you'll have more of these called CAMs. So CAMs and GAGs and proteoglycans are basically the glue that's between the cells, and it's made up of proteins and sticky carbs. Now, the fibers are embedded in that. So let's just look at some of these for a moment. And you can see the picture here, the proteoglycans. There's like one hanging out there. You can see these little green things that are showing you some of these CAMs and these GAGs and these glycoproteins. Now, the fibers, let me go to that slide. There's 3 types of fibers. So you're seeing the fibers here. Now, a lot of them are made up of collagen. Collagen is the most abundant protein in the body. It typically has a white color. There's over 20 different types of collagen. Okay, we're just going to put collagen together here. But collagen fibers are sometimes called white fibers. So if you think of a tendon or a ligament, so it's very, very thick. It's very strong, and it's inelastic. So elasticity does not mean stretching, okay? If something's elastic, it means if it's stretched, it goes back to its original size. Well, if you've ever stretched a ligament, had a sprain, you know it doesn't bounce back right away. So collagen fibers usually stain red or pink when you're looking at them in histology, and they're usually big and fat. Reticular fibers, on the other hand, are basically collagen fibers, but they're thin and branching. And I'll show you a better picture of reticular in just a moment. They're they form like internal scaffolding inside like the spleen or lymph nodes or the liver. Elastic fibers are formed from a different protein, which is called elastin. And just what it sounds like. Now, these are distensible and elastic. So distensible means to stretch, and elastic means to recoil. So they stretch and recoil. So they're going to be found in areas that need to do that. And if you take a deep breath, you'll realize they're going to be in your lungs, right? Now, junctions. There's 3 special types of junctions that you just have to kind of memorize what they are. And their name kind of tells you. There's tight junctions, desmosomes, and gap. All right, tight junctions. The cell membranes are interlocked like a zipper. This is what I was showing you with epithelial. So they're actually interlocked, and they're not going to pull apart. So when we're looking at these tight junctions, super strong. Sometimes they're called occluding junctions. I just call them tight. And think of the adjacent cell membranes interlocked like a zipper. They're impermeable. Now, desmosomes are kind of in the middle. They have a little bit of give. So if you think of a tight junction like a zipper, think of a desmosome like a snap, or you know those little metal clasps that you could hook on. So they're holding, but they have a little bit of give. So desmosomes are going to have a little bit of permeability. Now, as far as why you would have these, tight junctions are not going to let anything through the cell. Excuse me. So an example of tight junctions are in the cells that line your stomach. So your stomach lining, very tight junctions because you have hydrochloric acid in there. And you don't want that leaking out, right? You want to keep that in there. So you have tight junctions, it's impermeable. Desmosomes have some give. So you might see these in like your epidermis and your dermis where you have a lot of pull on that. Whereas gap junctions are actually ion channels. They're little pores between the membrane. So they're the most permeable. These are found in the heart, specifically in the heart muscle, because they allow the rapid flow of ions to just kind of pass right through the heart. Because when your heart contracts, right, it contracts as a unit. You don't want individual cells contracting. So that's the intro background stuff. Let's jump into epithelial tissue. So covering and lining. Everything you see on your body is epithelial, and the whole inside of everything is epithelial. The lining of your respiratory tract, your digestive tract, your urinary tract. It also forms the glands of your body. So why do we have epithelial tissue? Well, number one, protection. There's going to be cells that are going to be in many, many layers, right? Because if you want to protect, you don't just want one layer of cells. So we're going to have very many layers, and they're going to form a barrier for us. They also help with our immune defenses or our immune system. So some of the secretions, like in your sweat, are going to help protect you. Some of the cells in epithelial tissue are protective for us. And this ability to keep things out is one of the main reasons of protection, right? Because it helps keep pathogens from getting in our body. They also do secretion. There's going to be glands, as I mentioned, and glands are going to make specific secretions or substances. We'll get to that shortly. They do help move things through cell membranes. So these cells themselves are going to have different shapes that you'll see soon. And so they're going to have, they're going to be selectively permeable. So they're going to control what can pass, what can get through those cells for absorption and diffusion and secretion, as we mentioned. And they also can function a little bit with sensation. So your sense of touch, there's different receptors in epithelial tissue. So characteristics of this, a little more detail here. First of all, their cellularity. I said that they have tight junctions, so in sheets. A really good example of this is, have you ever peeled a sunburn? So as you peel that, it comes right off, right, in one sheet. That's because the cells are held tightly together. There's no matrix. It's just cells. Now, they have a top and a bottom. That's polarity. So there's an apical and a basal. So think B for bottom. The basal surface is the bottom. It's always next to connective tissue. The apical or apical surface is always the exposed surface. So when you're looking at this on a microscope or in a picture, the apical or apical surface is always going to face the white space, because the white space is going to be the opening, right? Or the top. There's nothing there. The basal surface will always have something underneath it, because that's where it attaches to connective tissue. And the way it attaches is through something called a basement membrane. So it might help you remember that the basement membrane is on the basal surface, and that's where the epithelial meets with the connective. I mentioned it's avascular. A in front of the word means that it does not have a blood supply. It does have a nerve supply, though, so it can supply and give you sensation of pain. And luckily, because it helps protect and it is so exposed, it regenerates really, really fast. So your epithelial tissue can actually regenerate and replace itself very quickly. In fact, in some places in your body, the cells only live for a few days. Your lining of your stomach, they say, is replaced every 3 to 6 days. So they have a very fast mitotic rate. They're stem cells, and their job is to always replicate and replace all those cells that die. So it's able to quickly replace dead cells. So how do we have to learn it? Well, there's 2 ways it's classified. So you have to look at the number of layers. Is it 1 layer thick of cells? Or, see, this is 1 layer of cells here. Or look at all these layers here. Or are there many layers? And so the first word is either going to be simple if it's 1 layered, or stratified if it's 2. Now, pseudostratified is 1 type only, and it means that it looks like it's stratified, but it's not. And I'll show you, you'll recognize that pretty quickly. Now, the other way we look at this is the shape. Is it flat, kind of like flat and irregular, like a fried egg? Then you could say squamous or squamous, potato, potato. Is it cuboid? Well, cuboid look kind of cube-shaped, but the best way to identify these is to look at the nucleus. So cuboidal tissue always has a big, round, fat nucleus. Or is it really elongated or columnar? And that has an oval nucleus. So the nucleus conforms to the shape of the cell. And if you just think about it for a minute, right? Structure determines function. A flat cell is not going to be really good at holding things. If I said, I'm going to give you a bunch of $20 bills and I want you to get a bag, are you going to get a little flat bag, or are you going to get a big bag? You're going to get something big. So the bigger the cell is, the more stuff it can hold, the more stuff it can secrete, right? Or the more it can bring in. So when you're getting into your columnar, they're probably going to be moving things more, secreting things. Cuboidal will also help, maybe not as much. And these squamous ones, they're not going to be secreting anything, right? That's not their job. So simple squamous is just one tiny layer of cells. Each of these cells here, there's one there. See all of these? You can see the nucleus. Those are all squamous cells. So there might be like, I don't know, 10 forming that circle. See the lumen? Very, very thin. So one layer of very thin cells is really, really good for moving things quickly, like diffusion. All right, that's how we want to move things quickly, through a thin layer of cells. So these are found in the linings of your capillaries, where you diffuse with your cells, and the alveoli, these are the, or alveoli, excuse me, these are the little air sacs in your lungs. So this is a superior view, which is why it looks different from this view. This one's a little more realistic, and you can see the cells. Sometimes they form what's called a mesothelium or an endothelium. Like in AMP2, we'll talk about the lining of capillaries, and I'll say it's called the endothelium, meaning it's just a layer of simple squamous. Okay, how about one layer of cuboidal? Well, what you're going to do is look for that big fat nucleus. See the large nucleus? That'll give it away for you, even if you can't see all the cell membranes. So these are roughly cube-shaped cells. And because they have more space, they're going to be doing more stuff. And in fact, what they tend to do is more secretion. So you tend to find these in your thyroid gland and your kidney. Now, the basic functional unit of the kidney, which you may know, is called a nephron. And you can see them really nicely there. And you also see them really easily in the nephron. So they're going to be secretion, in the thyroid gland. So secretion found in glands. Now, columnar is longer, so it's going to be able to store more stuff and hold more stuff. So these are the guys that we look at for secretion and absorption, mainly. Simple columnar often have microvilli. Microvilli are extensions of the cell membrane. So the cell membrane has these finger-like projections. They increase surface area. Well, it's good to increase surface area so you can have more absorption. So this is found in the lining of the small intestine. So if your small intestine is circular and food passes through the middle, it has to come in contact with the lining in order to be absorbed. So in this case, anything going through the middle doesn't get touched. But what if I add microvilli? Now everything passing through comes in contact with that cell. So the microvilli are going to increase surface area so that the nutrients come in contact with the cell and you can absorb more. You can pull that back from your lumen, the space, the hole in the small intestine, so you don't lose it to your large intestine and you can absorb it into your blood. Now, a lot of times you'll see these little clear white cells. These little clear guys are called goblet cells. And goblet cells are a special type of cell that makes mucus. So simple columnar, think of digestion system, small intestine in particular. Pseudostratified ciliated, this is the one I told you would never forget because it's got cilia, so it makes it really easy. And if you look, it looks striated, but they say technically it's not. I always think it's striated, but they say it's not. And it has cilia. So cilia, remember, circulate. Cilia are not microvilli. Cilia are little motor proteins and they move. And what they do is they help move debris and they move things past the cell. So they line your respiratory tract. So like your trachea, your bronchi, and what they do is they constantly move and they clean your lungs and move things up. And then it goes up to your throat, your oropharynx, and you swallow it. So when you swallow, you're swallowing debris from your lungs and cells. So they're really important in protecting our respiratory system. And as you would expect, lots of goblet cells, right, in your respiratory system making all that mucus. Okay, now stratified squamous or stratified squamous, look at all these cells and notice they look weird. The ones on the bottom look a little more normal than the ones on the top. And what happens is they start to get squished and they die as they move. So the ones down by the connective tissue are alive because remember, that's where the blood is. But epithelial is avascular, so these cells move, and as they move, they die. So everything you see on someone, the hair, the skin, it's all dead. It's too far away from the connective tissue. And so as the cells die, they get really flat and compressed. So many, many layers, which means it's going to be really, really good for protection. So this is one of the most widespread of all the epithelial tissues, and it's really important for protection. So places you would find this would be like inside your mouth, inside the vagina, the anus, the esophagus, the epidermis of your skin. So it primarily is going to protect because it has so many layers. Now, because we have this in different areas of our body, it can have keratin, which is a waterproofing protein. It basically helps prevent you from drying out, because if you, for example, this is in your epidermis. If your epidermis were to dry out, it will open and crack. So the keratinized is in the epidermis. The non-keratinized is found everywhere else. Now, I don't know if my lovely picture here is, let me see if I can, oh, I can move that. I never knew I could do that. So I'm going to move it up there. So you can see that the keratinized is in the epidermis, the non-keratinized is in other areas where you want to have protection. And then transitional, we just mentioned, you don't really have to identify it for me. It's kind of a weird tissue. It looks different depending on if you stretch it. So this is in your urinary bladder. It can compress and stretch as your bladder stretches and recoils. So those are the types of epithelial to know. Your book might have others. These are the ones I'm going to test you on. And I just want to talk about the glands real quick. So epithelial tissue forms glands, and there's two types of glands in the human body. There's endocrine and exocrine. Now, you actually kind of looked at endocrine last unit. Endocrine glands secrete their chemicals into the blood, and we call these secretion hormones. So a hormone is called a hormone because it travels in your blood to all areas of your body. And you've learned a bunch of endocrine glands: pituitary gland, thyroid, pancreas, etc. Exocrine is what we're going to focus on right now. And exocrine secrete through a duct. So a duct is like a little channel, and they usually secrete into like a body cavity or into your skin. So these are some examples: lacrimal for your eye, tear glands, a lot of your digestive glands, sweat glands, salivary glands. And you may have noticed that I said pancreas for endocrine. Well, pancreas actually has both. It has clusters of cells that make insulin and glucagon, but it also makes a lot of digestive enzymes. So sometimes an organ can secrete both ways. When we're looking at the glands, there's three main mechanisms of how they secrete. So how do these cells secrete something? So merocrine is the most common one. It's the one that you've probably most familiar with. You make the product, it goes into that little Golgi, goes in a little vesicle, and then it's just released from the cell. So most secretion is this way. Now, apocrine is a little bit different. Apocrine is when a little piece of it is pinched off, but it's very, very rare. So, in fact, I actually took it out of your notes. Holocrine is when the cell ruptures. So I like to think of it as the whole cell ruptures, right? Or you get a hole in your cell. So what happens is the secretions build up, and then the whole cell pops and it ruptures, and that's how it releases. Notice in merocrine, the cell is left intact. Now, merocrine is the main ones we're learning, but then they named them really, really weird. So I'm going to move my face down here now, I think. So when we're looking at sorry, my computer's being a little weird right now. When we look at sweat glands, there's two types of sweat glands. There's ecrine and apocrine, or apocrine. And I know you're like, I just talked about apocrine. I don't know why they named them that, okay? So we're looking at, this is the type of secretion. It's just going to be exocytosis from the cell. So your ecrine sweat glands are the most numerous in your body. When you think of sweat, that's probably what you're thinking about. And you'll notice that these sweat glands are going to have ducts. So they're going to secrete, usually right onto your skin. And they're very numerous in the palms of your hands and the soles of your feet. And so they're going to secrete sweat. And so sweat has a bunch of things in it. It's got a lot of water and electrolytes. It does have some urea. Urea, by the way, comes from, let me do it this way, ammonia. So NH3 is ammonia, and NH2 you might recognize as an amino group. So when you eat protein, it gets broken down into these amino acids, right? And then the amino group is removed, and it goes to your liver, which converts it into ammonia. And then that gets converted into something less toxic called ammonium, and then into urea. So we'll keep it simple. And then urea usually goes to your kidneys, and some of it goes in your sweat. So when urea sits for a while, it actually goes back to ammonia. So if you ever had like really disgusting shoes that you never wear socks with, or you have a gym bag that you have clothes in and you forget to wash your clothes and you leave your sweaty clothes in there, it smells really bad, right? It can burn your eyes out. That's because that urea has gone back into ammonia. So a little bit of urea in our sweat. And then we have something called dermacidin, which is basically an antibiotic or antiseptic. So it actually helps to clean your skin. So sweat is important. One of the main reasons that we sweat, though, is called sensible perspiration. We sweat to cool off. So anytime our thermoreceptors were too warm, we're going to sweat, and that's going to help get rid of waste, and it helps protect us. But the main reason is to regulate our body temperature. Now, that act of sweating is under control of our sympathetic division. You've probably heard of your sympathetic and your parasympathetic. So we have the autonomic nervous system, sympathetic and parasympathetic. The sympathetic is what activates your sweat glands. It releases something called norepinephrine, where we just say NE, and that's what's going to make you sweat. Now, another type of sweat gland, they call it apocrine. It is this merocrine secretion. Think A for axilla, if that helps you remember it. So these are in your axilla and your groin, and these are the ones that become active at puberty. And they're different because they secrete into a hair follicle. So they still use merocrine, they still use exocytosis, but it goes into a hair follicle. And so these are active at puberty, and they make a thicker sweat. So they make a sweat that's much more thick. It has proteins in it, and these proteins are nutrition for bacteria. So what happens is bacteria can hang out there. We got bacteria all over our body, guys, but they like to hang out in our groin, in our armpit, our axilla, eat this thick sweat, this protein, and then they make an odor, which causes body odor or BO, and that's where that comes from. Now, two other examples, they're actually in the integumentary unit, but I'm just going to cover them now. Mammary glands and ceruminous glands. So mammary glands are under a hormone called prolactin, or we say PRL, and you probably know they produce milk in mammals. That's how we feed our young. And ceruminous glands are going to make cerumen, or cerumen, again, potato, you'll hear it both ways, which is earwax, which is there basically to protect you and prevent things from climbing in your ears. Now, as far as the holocrine, right, the whole cell ruptures, this is what releases sebum. So they're called sebaceous glands, and sebum is oil. Now, sebum, again, helps inhibit bacteria. It is oily. It helps protect the keratin and keep your skin and your hair lubricated as well. So it does have a function. In fact, in fetuses, as they're getting toward the end of their development, the sebaceous glands get very, very active, and they make this waxy coating over the baby so it can just pop out. It doesn't always pop out like that, but it's called the vernix caseosa. And so this is just like this layer of sebum, and it helps, again, with a vaginal delivery. So if we're looking at epithelial tissue, this is from your book. I think it's a nice overview going over the tissue. I think a lot of times you can learn a lot by looking at an image. So maybe take your time and read over this and look at the pictures. And then I want to just show you this image as well, just to real quick talk you through how do you identify tissues. When you're looking at epithelial tissue, remember what you're looking at is the cell shape and the number of layers. In fact, I think if you just look at the nucleus, you can figure it out almost every time. So notice that the nuclei here are kind of short and fat and round. Anytime you see round, you know it's cuboidal, and keep it easy. You're only learning simple cuboidal. You're not learning stratified. But here you can see, if I draw a line, see the white in the middle? So that's the apical surface. Notice here, I see lots and lots of scattered flat cells and nuclei. So that tells me if it's flat, that squamous or squamous. But look at all these layers. So I know that this has to be stratified. So as you're identifying the tissues, check out the nucleus, and it'll help you figure out what the tissue type is. So that finishes our lecture on the epithelial tissue.